Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
He started out actually studying the olfactory system at Caltech in the CNS program. And something about that thesis had a permanent impact on the field of computational neuroscience. He had to write a simulator to simulate biophysically realistic compartmental models of neurons. And that simulator became Genesis, which is a package that's now routinely used by many labs to simulate networks of spiking neurons. But he went on from there to the University of Arizona, where Bruce McNaughton was developing techniques to increase the number of neurons he can record from. And in fact, I confirmed this with Bruce McNaughton. Their paper that was published in Science was the first paper that achieved simultaneous recording from 100 neurons at the same time. So that was a breakthrough. And in fact, it wasn't until recently that a new technique, optical recording, has emerged that allows us to take that up to thousands of neurons. But in any case, you'll be hearing today from some really groundbreaking work. Tremendous progress is being made, and a lot of it's coming from Matt's lab at MIT.
I can use the podium mic. Yeah, it's going to be a little tough being stuck to the podium. I like to move around a lot. One of the things I'm going to be talking about is rodent spatial navigational memory. Oh yeah, I'll do it. Great. And I was telling Terry, actually, I think the last NIPS I came to was NIPS 1, 1987. It's 26 years ago. I was sitting out there just like you, and it was it was really inspirational for me as someone who was really coming into a new field. My background was in engineering. I was trying to sort of crack into the world of neuroscience, but bring with it ideas, principles from engineering, information theory, thinking about the system as a computational entity. And NIPS for me was really transformative. So I'm hoping that perhaps I can have some influence on some of you to get you to think about sorry, I just noticed there's nothing up on the screen. Oh, there we go. Let me just try this. There we go, good enough. Well, think about biological systems, and in this case, the hippocampus, as potentially a source of inspiration to give you ideas for ways in which computational ideas that have been manifest in the biology, but perhaps have not been evident. And as Terry pointed out, when I sort of moved into the world of neurophysiology, developed techniques for doing recording, large-scale recording of many individual neurons. Can I get a pointer?
Up there, yeah.
Got it.
Top button.
Really the idea, I had started working in the olfactory system. The idea was I'd come over to the hippocampus. It was an interesting system. Bruce McNaughton had been using it to study the expression of behavioral correlates in freely behaving animals. I'd always imagined I'd go back to the olfactory system. I still think that's possible. We were actually just talking, Terry, Pierre, and I were talking about sort of new ways of thinking about the vision problem, perceptual problems. What does it mean to actually to see the world? And you can say the hard problem that the hippocampus really has to tackle, and all systems have to tackle, is how do you build models of the world through experience? And so what I'm going to try to describe is the work that we've done recording in this structure, the hippocampus, to look at the development of internal representations of space as animals experience the world. And one of the working hypotheses that I'm going to try to advance is that this notion of memory, in particular in the hippocampus, is really embedded into this notion of experience, and that is that damage to the hippocampus in rodents impairs spatial memory, spatial navigational memory, moving through the world. In humans, it seems to impact episodic memory, event memory. Both of these things are linked in the sense that they require that one evaluate not simply events, but the temporal linkage of events. The time in evaluating the time course of events becomes critical. Of course, when you have time, time gives you the ability to evaluate not only where you are, but where you have been and where you will be. It embeds within it a system, a structure for doing adaptive predictive coding. And hopefully, I will be able to, again, inspire you to think about how the biology has come up with clever ways of doing this. The device that Terry had mentioned, shown here, this is a recording device that houses a number of electrodes, an electrode array, allowing fine microelectrodes to be advanced into the brain. This is implanted chronically. It stays in the animal's head. These electrodes, when they actually go into the brain tissue, sit outside in the extracellular space near neurons where they can record the discharges of collections of neurons. And I'm just showing you this little scatter plot, which illustrates the raw data we actually get from the system. Each point here is an action potential recorded from a collection of 4 wires. In this case, we're looking at the projection on two of those wires are 2 channels in which you see the amplitude of an action potential here displayed. One point here, for instance, has a high amplitude on this channel, smaller amplitude on this channel. This essentially gives you a 3-dimensional triangulation of neural spaces. This is the distribution, spatial distribution around the electrode. We put in 10, 20 of these electrodes, and so we can record simultaneously a large population. Now, one of the things that was attractive about the hippocampus when I got into this was the observation that had been made in the early 1970s by John O'Keefe, who really did the seminal work in the hippocampus, in which he noted that when animals are allowed to experience the world the way rats experience the world, that is, moving through space, individual hippocampal neurons express this behavioral correlate known as spatial firing or place fields. This is an illustration of the spatial firing or place field phenomenon in which the color coding represents different hippocampal neurons. Individual points or individual action potentials. And what you're seeing here, this is the cumulative activity over about 10 minutes of about 8 to 10 cells. The yellow cell here, for instance, you see this is a top-down view of a little C-shaped track about 1.5 meters this way. And when the animal walks along this path, you can see this yellow cell discharges robustly here. The red cell fires here, the green cell fires here. Different cells fire in different locations on this track. Now, one thing about these cells that O'Keefe noted was that while animals that explore in an open field, randomly foraging, the cells will fire based on the animal's location. When they follow circumscribed paths, for instance here on this track, in other words, when they move through space in systematic sequences, the cells develop this property that was referred to as directionality, and that is they'll only fire when the animal moves through this region of space in this direction. The yellow cell fires when the animal goes through here, pop, 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 pop. But when it comes back in this direction, it doesn't fire. It fires in a different fashion. So it's dependent not only on the location, but the direction. Now, you can think of directionality as simply being, that's a single cell version of trajectory dependence. And that is that when the animal walks along this path in this direction, you'll get a yellow cell, red cell, green cell. That would be a sequence. When it moves back through this in the opposite direction, you don't get the reverse sequence. You get a completely different sequence. So there's a sequence or trajectory encoding that is at least it's possible given this directional dependence. And I'm just going to show you, this is some raw data. This is raw recording taken from an experiment in which an animal is walking along a 10-meter track. This is this display. The cumulative display here is showing about 6 cells, color-coded. And what you're seeing here are the action potentials as they're being produced, where the animal is. The green circle represents the video tracking of the animal. Now, a couple of things to know. One is you see the spatial bias, blue cell fires here, for instance, light blue cell fires here. But if you listen really carefully, you can hear a couple of things. One is that there's an oscillatory modulation, rhythmic modulation of activity when the animal moves. It's a 10 hertz oscillation. It sounds kind of like this. Now, it's gone now because the animal's stopping. Can you hear that? I guess I have tuned ears, so it just jumps right out at me. But it's very characteristic of this when the animal's moving. When the animal stops, there's a change in the mode of activity. The rhythm goes away and it's replaced by this kind of aperiodic bursting, this oh. Let me just do one more time. I'm not going to do the whole thing. But I want you to listen when the animal stops. You'll hear this bursting, and you'll see many cells firing. See, here's the spatial firing. There, you hear that? That's an aperiodic. It's just a lot of activity. Many more cells fire than fired during actual behavior. While the animal's moving, on average, about 1 to 5% of the cells are active. During one of these short bursts, 30%, 30 to 50% of the cells can fire. So it's this very, very energetic period during which you see lots of cells responding. What I'll show you is that when we zoom in on those events, those bursting events, we find that the activity there is not random. It's actually highly structured and structured in a very interesting way. So that's just showing you the raw data. That's what you would see if you were sitting in the rig, you're watching the displays, you'd see these spikes coming out. But we can go back afterward, post-process the data, analyze patterns of activity across the full neuronal ensemble of maybe 100 cells, and we can do some simple sort of Bayesian decoding so that we're going to estimate. Now we're going to estimate using the posterior distributions derived from our measurement of the spatial biases during behavior. So we have the blue cell fires here, light blue cell fired here. So now we're going to go back and in 100 millisecond intervals ask which cells are firing and then simply estimate the probability that pattern would be generated given that the animal was in any one of the possible locations along the track. And so what you're seeing here is that the blue triangles, they represent the probability that the animal is in that location given that you see a pattern of activity within a 100 millisecond window. And what you see is that while the animal's moving, the triangle tracks position pretty well. When the animal stops, you see that estimate breaks down, the triangle jumps all over the track. And so these two modes, the animal's actively exploring, you see this rhythmic mode, referred to as the theta rhythm. It's this 4 to 12 hertz oscillation that very quickly transitions into this aperiodic bursting, referred to as these sharp wave ripples. Sharp wave, because of the highly excited state of the hippocampus, this burst of excitation driving a lot of neural activity shows up here in the extracellular field. So this is the field potential, the electrical field generated, the average, you can think of as sort of the average macroscopic electrical activity in the vicinity of these electrodes, in which you see this oscillation, and then you see these little bursts, these sharp spikes of activity. Now, it's in these sharp spikes of activity that, I'm going to refer to this state as the offline state. And one reason for that is that this transition that occurs when the animals when they stop moving is also what you see when the animals enter into early stages of non-REM sleep. So this is what the hippocampus looks like when the animal goes to sleep, as well as when it simply stops. Now, one thing to note about this, the activity during locomotion, during movement, is that this notion of a spatial representation. So I showed you this decoding where we can take ongoing population activity every 100 milliseconds and just make an estimation, probability that the animal's in a given location, decode position. But if we take that window and we shrink it from 100 milliseconds down to 20 milliseconds, so now we're doing very high resolution decoding. So here, this is decoded position. It's a function of time. This is just over a second. Here you see this oscillation, the theta oscillation. And now we're going to be doing much higher temporal resolution decoding. And what we see here is that the decoded position is actually not stationary. In fact, it varies as a function of phase within this oscillation, such that the estimate, the spatial estimate, is in fact sweeping from a position just behind the animal to just in front of the animal 10 times a second. So the actual representation of space as the animal's moving is in fact a moving trajectory itself that goes from where the animal was to where the animal is going to be. There is this retrospective prospective sweep. So if I were to reconstruct this is what the hippocampus is actually computing, you could say, 10 times a second. It's an estimate, a moving estimate that kind of sweeps from behind it to in front of it. This kind of this is the representation of space that the hippocampus has. It's not stationary. It has this sequential component to it. Now, this is going to be relevant because we're going to see that this time scale, the time scale of a single theta cycle, roughly 80 to 100 milliseconds, is also the time scale during which we will see what I'll refer to as the reactivation or the replay of prior memory in these small 100 millisecond packets, suggesting that there is sort of a discrete time scale over which brief sequential information is encoded and then subsequently processed. And it's the time scale of the theta oscillation. Now, we've done a lot of work trying to explain, model, otherwise understand how this sequential representation comes about. I'm not going to talk about any of that. We just take it as a given. Hippocampus represents space as a set of short sequences. So now we have some understanding of what the hippocampus is doing while the animal is actually moving through space. But the idea of spatial memory, and again, I'm trying to connect the notion of spatial memory as memory of spatial trajectories, with ideas of event memory, episodic memory, memory of events over time. And to understand how memory might be maintained and expressed is difficult to do it during behavior. Problem is that you have behavior, you have a behavioral dependence. But sleep gives you an opportunity to do three things. One, it allows you to examine the structure and content of the underlying neural substrate independent of the external world. So you could say during sleep, any structure that you see must be the consequence of memory. You can say it is almost by definition memory because it is it's the residual structure from experience. It's not being driven, simply being internally expressed. Second thing is that there have been sort of theoretical and also empirical suggestions that sleep may contribute in some way to memory processing. There might be a role for sleep in memory. And we can break down sleep broadly into two different stages: rapid eye movement sleep, which you tend to think of as dream sleep, even though that's somewhat of a misnomer, the idea that there's dream and non-dream sleep. Dream sleep, and that is sleep during which there is some kind of structured internal mentation occurs during all stages, including the sort of the phylogenetically old type of sleep, non-rapid eye movement sleep, which, as I pointed out, bears close resemblance to the kind of activity one sees during quiet wakefulness. So simply sitting quietly and thinking looks a lot like, electrophysiologically, like non-REM sleep, leading some very interesting models, such as the fly sleep models. Fly sleep bears a lot of similarity to mammalian non-REM sleep, which bears a lot of similarity to general vertebrate quiet wakefulness. So I'm going to focus on non-REM. We've done work in REM sleep. I'm not going to talk about any of that. So if we just think about non-REM sleep, that's the early stage of sleep that you would enter into now. If I wasn't jumping around, you started getting bored, you started to nod off, non-REM sleep is what you're going to start to drift into. And so we can look at that. You can see here the structure. This shows sleep structure prior to and following a period of active behavior. So the animal runs for about 15 minutes. We take him off, put him on a little platform, and he gets bored and he starts to go to sleep. And you can see here in the light green, these are periods of non-REM, or largely slow wave sleep. Slow wave sleep refers to the synchronized oscillations that one sees primarily on the surface cortex. If I would just put EEG electrodes, what I would see is these slow oscillations that would start to emerge. And this is the first stage of sleep that you'll enter into. Once you go into some slow wave sleep, then you'll start getting periods of rapid eye movement sleep that will become more frequent. And longer in duration, the longer you stay asleep, until you get to the period here. This would be the period of sleep offset. If I really put you to sleep and you stayed in here for 7 or 8 hours and you didn't wake up until tomorrow morning, this is where you would be right here. This is the period of sleep offset after you've awakened from an extended period of sleep. And that tends to be more heavily loaded with REM, rapid eye movement sleep. A mammalian innovation, which we could discuss afterward what that what actually means, but again, I'm not going to talk about that. So we have running, and we have the animal going right to sleep. And Albert Lee, when he was a graduate student in my lab, looked at this period of non-rapid eye movement sleep immediately following behavior. And what he noted was that if you look at activity during active behavior, here are 8 place cells. And in this case, they fire in a sequence. Place cell 1. These are ordered so that you can, based on the location of the peak of their place field. So place cell 1 fires here on the left-hand side of a track. This is position here. The animal is going to run from left to right. Place cell 1 will fire first, then 2, 3, 4, 5, 6, 7, 8. So when the animal runs from left to right, you'll get a sequence from 1 to 8 that takes about 5 seconds to run through. That's how long it takes the animal to run through. When the animal would go to sleep, following that, there were these periods, brief periods lasting roughly 100 milliseconds or so, in which you saw the same cells firing in the same sequence. And as they preserve the order, but not the time scale of sequential activation. And that these brief, ordered sequence events coincided with the expression of these sharp wave ripple events, these little bursts. So that when you were listening, you heard that shh. That was one of these events. It's a brief high-frequency oscillation, that's referred to as the ripple, during which the population expresses its activity in this sequential fashion, recapitulating prior behavioral sequences. So this was in the hippocampus. Now, as I pointed out, there's some ideas about sleep and its involvement in memory. And one of those ideas that it participates in a process that's been sort of loosely referred to as memory consolidation, and that's the transformation or transferal of information following from a short-term store into a longer-term store. Reorganization, synthesis, but something that takes a memory of recent experience and turns it into memory of long-term experience. And many of those models involve an exchange of information between different brain structures. And one of those target brain structures that's been postulated is the neocortex. The neocortex, where that's our interface to the world. If there is a world model, it probably exists largely out in the neocortex. And so one question is, if we see these events occurring during sleep in the hippocampus, is there anything corresponding to these events in the neocortex? Diane-G, when he was a postdoc in the lab, did some work recording simultaneously in the primary visual cortex and in the hippocampus. And one of the other motivations for doing this particular experiment is this idea that do rats dream? Dreaming being something that we subjectively experience, but it brings with it not only the recapitulation of some perhaps memory-inspired, temporally structured information, but there's also a perceptual component. You see what you dream, you experience it in a way that corresponds, in some sense, to your actual experience. So the question is, do rats also have a perceptual counterpart to these reactivation events? And so the experiment involved simultaneously recording in visual cortex and in the hippocampus, and then having the animal run. It's just a variant of one of these little tracks. We refer to them as mazes. In this case, the animal's just running a little figure 8. One of the changes that we introduced into this, it's interesting, given the renewed interest in the rodent visual system. We had tried to do this experiment years before this, trying to record in the primary visual cortex of rodents running in visual environments. The problem that we had is that we had used a primate-centric model for vision. That is, we used large frontal field presented visual cues. We found very poor response in the visual cortex, very poor discrimination. Different stimuli, different patterns. Visual cortical neurons didn't seem to really differentiate between them. But Diane-G, the postdoc who did this experiment, had the insight that if we're going to do experiments with rats, perhaps we should think like a rat instead of a primate. And so the innovation here was to take the stimuli from out there and bring them down into the proximal lower visual field where the rat's world generally is. That's where they live. We live up here 2 meters off the ground. The rats live down there, about 2 millimeters off the ground. And so down in that world, these cues dominate. So when G put the visual cues for the rodent down in the proximal lower visual field, what he found was now over 70% of the cells actually are visually responsive. They're highly discriminative, and they exhibit what was a surprising property. And that's illustrated here. This is just a single visual cortical cell. This visual cortical cell seems to show a spatial receptive field, and that is it fires in this case, a circumscribed region of the tract. Despite the fact that visual cues are repeated, so they're not unique visual cues on any given location of the tract. They're unique configurations of cues. In this case, there were 8 distinct visual patterns, and the configuration of patterns would vary. But in this case, the visual cortical cells showed a spatial preference. And what that allowed the same kind of analysis that we had performed in the hippocampus, and that is, in this case, I've ordered about 8 of these visual cortical cells, and so you can see during running, there's a visual sequence. This is the sequence of visual cortical response that occurs as the animal moves along a trajectory in this track. So here you have a visual sequence that occurs during running. Here in the hippocampus, we have this spatial sequence that occurs during running. And so now we can ask the same question. When the animal goes to sleep, do you see evidence of sequential reactivation in the hippocampus and in the visual cortex? And do the two actually align? In other words, when the hippocampus is expressing a spatial sequence along this path, is the visual cortex expressing a sequence, a visual sequence along that same region of the tract? I'm not going to show all the data. This is published, but the answer is yes, that the visual cortex does recapitulate perceptual sequences that are coordinated with the underlying hippocampal sequences. And just one thing I want to point out about this in the study, we noted that when you look at the macroscopic activity, you step back and say, well, what's the visual cortex doing? What's the hippocampus doing on a longer time scale? What you see here, now, this is several seconds of activity during sleep. And here you see the manifestation of this slow wave, the slow oscillation of slow wave sleep. This is a slow fluctuation in excitability in the neocortex. Here you see periods of heightened activity interspersed with periods of inactivity, silence, active silence. Now these would correspond, if we went in and recorded from individual neurons in the neocortex show these transitions between depolarized and hyperpolarized states, so-called up states and down states. They flip on and they flip off. And so during sleep, you have this kind of, it's like this shh, 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 shh. This is the slow wave of slow wave sleep. And during these up states, neocortical neurons are synchronized. They're all turning on, they're all turning off. It's this synchronized on and off transition. And what G noted was that these transitions, up-down state transitions in the neocortex, they had a phase relationship with up-down transitions in the hippocampus. They lead it. Neocortex turns on, then about 80 milliseconds later, hippocampus turns on. On. Neocortex turns off, hippocampus turns off. So the hippocampus seems to be following the neocortex in this case, in these up and down states. But when G looked at structured activity, so these sequence reactivation events, when they occur, they're occurring during these up states. They're occurring during the expression of these sharp wave ripples. So here, this is the extracellular field, the electric field around these electrodes in which you see these high frequency oscillations, the sharp wave ripples. And one of these upstate periods lasts about 500 milliseconds. One of these sharp wave ripples lasts about 100 milliseconds. So what's happening during one of these upstate periods is that you're getting a burst of ripple events. You're getting a series of short 100 millisecond events. This is where these sequences are being expressed. And when you look at the lead-lag relationship between the sequences, now you find that the relationship reverses, and that is the hippocampus leads. So the neocortex leads in starting it off, but the hippocampus leads in expressing the sequence. So what it suggests is that there's actually this structured dialogue. The neocortex kind of starts things off. It says, that little circle pattern, sends the circle pattern down as an initial state to the hippocampus. The question the neocortex wants to know is, do you have any memory of events that occurred associated with this circle pattern? The hippocampus, oh yeah, circle pattern. That was when I was running along this, the left-hand loop. So initiate with circle pattern, and then express the spatial sequence associated with that. When the hippocampus expresses the spatial sequence, that sequence then goes back to the neocortex, and the neocortex can now reconstitute the perceptual components that were associated with that spatial sequence. So that's how the neocortex gains the ability to express these perceptual sequences by leveraging the hippocampus. Initiate and then respond. Now, this idea, the idea that the neocortex somehow establishes the initial condition for hippocampal reactivation, is something that another postdoc, Dan Bender, in my lab, decided that he was going to try to exploit. And so the idea is, gee, if the neocortex can bias these neocortical upstates, or whatever the neocortex is saying during these upstates can bias hippocampal reactivation, what if we can bias the neocortical upstates? What if we can just provide a sensory input to, in some way, bias what the neocortical sensory neurons are doing? We should be able to gain the ability to bias these reactivation events exogenously. And so he did a simple experiment. Trained an animal to run on this instrumental task, and that is the animal sitting here, pokes its nose, and then he hears one of two tones. They're actually tone sweeps, up sweeps or down sweeps. So the spectral content is identical, they're just different sequence, going up or going down. But they're two different tones. You hear the up sweep, run to the right. You hear the down sweep, run to the left. And that's it. Animal just learns to do that. Hear the right sound, go to the right. Hear the left sound, go to the left. And then we let the animal go to sleep. You just take the animal off the task and it goes to sleep, and we keep playing the sounds. And we ask the question, while the animal is now sleeping and hearing these sounds, and now we decode, we look at these reactivation events, are these sounds influencing some way what the hippocampus is actually reactivating? So here, this is the task. The animal is running, the animal is sitting here, he's sitting on this point. Play the right-hand sound, he runs to the right-hand side, he comes back, you play the left-hand sound, he goes to the left-hand side. And there are place cells that fire on the right, place cells that fire on the left. And so this is the experiment. The animal's awake, we're playing these auditory cues, left sound, right sound, the animal's running the task. Then it goes to sleep, and we keep playing these sounds. This is just one, this is a little 750 millisecond sweep, a little wheep, phew. That's what he hears, wheep, phew. But once every 5 to 10 seconds. So it's not, not unlike what you might actually be hearing if you're you're asleep. And like this morning, I hear these, I hear the backup tones from the trucks out in the parking lot. It's like every 10 seconds I hear this stupid backup tone. Did it influence what I was dreaming about? Well, that's what we're actually doing here. We're giving tones that have some particular spatial correlate and ask whether it biases reactivation. And so now, here, this is during sleep. We play a sound. We look at these during these sharp wave ripple events. We're going to decode the content, and we're going to ask, when we play the left-hand sound, do we see place cells on the left-hand side being biased in their reactivation? And I wouldn't show you this if that wasn't the case. And so that was the result. The result was, play the left-hand sound. When you play the left-hand, you play the left-hand sound, you get left-hand, you bias left-hand place fields. Play the right-hand sound, you bias the right-hand place fields. And this just shows it for individual units. Now, there's one interesting thing. I'm not going to show you any of the data on here, but after I talk about some of the sleep stuff, I'm going to switch to quiet wakefulness, because as I said, quiet wakefulness looks a lot like sleep from the hippocampal perspective. But there is one difference. Well, there are probably a lot of differences, but one key difference is that during sleep, not only is the hippocampus offline, but the neocortex is also offline. It's a state, the slow oscillation is essentially a state in which you have this kind of thalamic decoupling. Neocortex is now offline. During quiet wakefulness, you could be sitting here, you could be the hippocampus could be, you could be thinking about, oh, what am I going to have for, what did I have for breakfast this morning? What am I going to see for dinner tonight? So you can be thinking about other things while simultaneously you are still perceiving the world, presumably so that you can respond appropriately. You might not be remembering any of it, but you're still responding to it. So the neocortex, during quiet wakefulness, the neocortex is online. So we did exactly the same manipulation during quiet wakefulness, and this doesn't work. We can't influence reactivation or bias reactivation during quiet wakefulness, doing this only during sleep. And now another interesting thing about this bias is the tone itself is 750 milliseconds. But as I said, these tones were occurring every 5 to 10 seconds. When we looked at reactivation following the tone, the bias would persist for many seconds. So the cue itself wasn't driving reactivation. It was simply biasing a state, which was then biasing reactivation. So even 5 to 10 seconds after delivering that tone, the hippocampus was still biased in its reactivation of those. You play the left-hand sound, and for 10 seconds, you're thinking about running on the left-hand side of the maze. Again, suggesting that there's some persistent influence that's initiated by the cortex, but then propagated through some kind of dialogue or interaction. Now, this is compatible with a number of behavioral psychophysical experiments done in humans showing similar effects. Sleep cueing of memory leading to behavioral performance. In this case, some work by Jan Born doing this with odors, and then in Ken Paler's lab at Northwestern doing the same sort of thing with sounds. And in this case, in this case, subjects were trained on a particular little spatial matching task, match some cards. They do it with an odor during sleep. You give the odor again. When they wake up, they're better at doing the task if they had the odor than if they didn't. And they're better if they were given the odor during non-REM sleep. In this case, it's the same idea. They're given tone associations, though. You know, match, find the cat cards. But when they would flip over the cards, they would hear the sound of a cat. So you hear the meow, you see the cat. Then you have some, the elephant cards, you hear the elephant. If you just play the cat sounds during sleep, they get better at the task, but only for the cat cues. So it's selective enhancement, cue enhancement during sleep of specific information. Now, the idea is then that what you have is that our stimulation, this cueing, influences the cortex, which biases this initial state, which then influences what the hippocampus reactivates, which then goes back and influences what the cortex expresses. I'm getting squeezed on time, so I'm going to now switch over to this other offline state in which we see the same kind of reactivation event, and that is during quiet wakefulness. So this example that I gave you, when the animal stops, within about half a second, the hippocampus switches into this offline reactivation mode. And during quiet wakefulness, we see activity that looks very much like non-REM sleep, bursts of activity. The question is there any structure in that activity? And an experiment by Dave Foster when he was in the lab, that's a very simple question. Animal running along a track. Now, when animals run these tasks, they run. There are periods of this theta rhythm. You see place cells. And then they stop. They stop and they receive food reward. These sharp wave ripple bursts, the quiet wakeful state, occurs when animals are engaged in what's referred to consummatory behavior, when they're satisfying internal state demands. They're eating, they're copulating, they're grooming. It doesn't make any difference what you can be thinking of anything, right, when you're doing these. Are automatic behaviors. The hippocampus switches into this offline mode. And so when the animal stops, eats, what's it thinking about? What Dave Foster found, this is just the raw data. This is a single trial now. This is the position of the animal. The animal starts at one end of the maze and runs to the other end of the maze, and it just sits there. It just sits there for like 2 minutes. It doesn't do anything. Now, the reason this is data at all is because Dave Foster was willing to let an animal just sit on the track doing nothing for 2 minutes, which most people in the lab would not do because that's 2 minutes of nothing. You're not collecting any data. The idea is, no, we need the animals moving. That's when you see place cells. That's when memories are being encoded. But Dave Foster, by allowing the animal to sit, was able to see what occurred during these periods of quiet wakeful reactivation. And here I've just zoomed in on one of these events. I zoom in on one of these events, and this lasts about 500 milliseconds. You see these high-frequency oscillations to sharp wave ripples, and then you see the individual, in this case about 20 individual place cells. And they've been ordered. In this case, place cell 1 fires on the left-hand side of the maze. Place cell 19 fires on the right-hand side of the maze. So when the animal runs from left to right, the place cells fire, in this case, in order from 1 to 19. But during this event, the place cells are firing in order, but in this case, the order from 19 back to 1. They're firing in time-reversed order. It's time-reversed reactivation. Now, there are a number of questions that came up from this. That is, well, what happens if, is this something that's associated with reward? What would happen if you were in really large environments? Like, it's one thing to think about memory reactivation, a little track lasting about 5 seconds, but what about just moving around in the world, seconds, minutes, hours, extended sequences? And this idea that sequences can be expressed in reverse, I showed you these sequences during sleep which seem to go in the forward direction. So is there a dichotomy between reactivation during quiet wakefulness and sleep? You see forward sequences during sleep, reverse sequences during quiet wakefulness. Well, to follow up on this, another postdoc in the lab, Fabian Klusterman, and a postdoc, Tom Davidson, redid that experiment, but now using a much longer maze. This is a 10-meter long maze. It takes the animal about a minute to actually run along this track, and it will stop at various locations. And so we get to look at reactivation of extended sequences, reactivation that occurs at points of stopping that don't involve reward, and when the animal stops in the middle of the track, there's the possibility of both forward and reverse sequences being expressed, which was a complication in the previous, the end-to-end maze. When you stop at the end, a forward sequence there really isn't a forward sequence, it's all reverse. So there was a confound there. So what they found was that yes, there are extended sequences, and they can be expressed. For example, here when the animal stops in the absence of reward, but they found something else that was really interesting. That is, extended sequences are formed by concatenating a number of these short sequences. These short sequences are the length of a single sharp wave ripple event, this 100 millisecond sequence. So longer sequences are composed of shorter 100 millisecond sequences. And I'm just going to show you one of these. So this has been slowed down by about a factor of 4. I'm just going to turn down the volume a little bit. Factor of 5. And so this is, again, about 500 milliseconds, the entire event. This is the decoded position. Now, in this case, taking it at 20 millisecond intervals. And what you see is the decoded position follows a trajectory that moves from where the animal is to, in this case, about 5 meters down the track. Now we see reactivated events like this that go in the forward direction, the reverse direction. They can be initiated at remote locations, so it's not just where the animal is. Animals can think about other tracks. If there are other tracks that the animal has been on, sequences can be expressed in other environments. And that they span these multiple ripple events. So it suggests this compositional constraint of sequences. They're built with shorter sequences. Now, these shorter sequences, I already mentioned to you a mechanism for encoding shorter sequences. It's these theta sequences. So the hippocampus is already encoding experience in the form of these short 100 millisecond sequences. And the sequences that you see re-expressed, again, are the same time and spatial scale as these theta sequences. The difference is that the theta sequences, as the animal's experiencing them, overlap. The reactivated sequences, they don't. They're these sort of piecewise continuous. You can think of them as the optimal ordered sequence. They throw away absolute time, but they maintain time order. Now, this idea that you have sequences, these reverse sequences in particular, is something that inspired Dave Foster, who had worked with Peter Dayan working on reinforcement theory models of navigation. And his first thought was, hey, this reverse sequence reactivation could solve the temporal credit assignment problem. I'm down to about 1 minute. So, and the idea is you can solve the temporal credit assignment problem by playing a reverse sequence. At the same time, you deliver a reward signal that has this particular graded form. If you actually convolve this kind of discrete reward signal that would occur when the animal's at the end of the maze with this, in this case, this kind of graded reward signal during a reverse sequence, you can turn this discrete reward signal into a spatially graded reinforcement signal in one shot. Solves temporal credit assignment problem in one shot. But it requires that you replay sequences along with a reward signal. And that you could use this to guide navigation, to learn spatial sequences. And just recently published some work showing that that during a guided navigational task, these replay sequences actually predict the animal's future behavior. They are being used in some way or reflect the animal's future planned trajectories. So I'm running out of time. I'll quickly close with some new unpublished work that addresses that question, the hypothesis that reward signaling could be paired with these reactivated sequences to solve the temporal credit assignment problem and solve a reinforcement learning problem that the animal might have. And this is done by recording dopamine activity, dopamine cells, in the VTA, ventral tegmental area, and the hippocampus while animals run in that simple spatial working memory task. So the animal's going to run a little task where it starts at one arm and then has to choose one of two arms. And the choice is informed by where it started. It starts on the right, has to go to the right. When the animal comes back, we force it into one of these two arms. So in this direction, the animal chooses. In this direction, we choose. So we force it into a starting arm, then the animal has to make a choice. And in the chart, what we see, there's reward signaling that occurs while the animal makes a nose poke. In this case, you see here's dopamine activity, dopamine cells, time locked to reward delivery. You see cells that fire during delivery of reward. You see cells that fire on approach to reward. And if we average all this, you find something that's interesting in that when we model this, we can model the development of these dopamine responses based on a simple QTD model. And in this case, we see dopamine activity that's generally flat and then begins to ramp up. We see these ramps of dopamine response following the choice point, and that the activity predicts incorrect or correct choice. And that if we ask, do these dopamine cells, are they time locked in any way to these ripple events? So we have hippocampal single unit activity. These are the bursts. These are the ripples in the hippocampus. These are dopamine units. And if we now time lock, we look at the activity of dopamine units relative to these sharp wave ripple events. These are hippocampal units relative to ripples. You see, yes, hippocampal units follow sharp wave ripples. Dopamine units also follow sharp wave ripples, but we can go an extra step. We can decode activity. We'll decode these replay sequences. And so we can identify the hippocampus replaying a sequence along the track. And maybe 30% of them, we can decode. The animal about 30% of the time is actually thinking about running on the track. The other 50, 70% of the time, thinking about something else, getting off the track, having lunch. And so we can distinguish those. So here, for instance, is an example. Here's one sharp wave ripple sequence, about a couple hundred milliseconds, consisting of a trajectory that decodes along the track. Here's another sequence that we can't decode. And it's something, but it's activity. It's just not decodable activity. And now when we ask, if we look at dopamine activity relative to these two types of sequence based on the content, if we look at replay sequences, yes, indeed, dopamine units, they lock to it. If we look at non-replay sequence, just as much hippocampal activity, but the content is not replaying anything on the maze, now the dopamine units are not locked to it. So these dopamine units are sensitive not simply to the event, the sharp wave ripple event, but to the content. And I'll just flip over that last thing. The units that lock to the sharp wave ripples are the same units that lock to the theta rhythm. So the theta rhythm, again, the encoding event, is a correlate that hippocampal units will lock to, dopamine units will lock to, and that predicts their participation in these ripple events. Now, we finally decide, okay, let's look during sleep. When we look during sleep at the same thing, what happens? It's all gone. So we had this grand idea that yes, during sleep, you're sort of recapitulating learning, you're reactivating memories, you're reestablishing associations, but this dopamine unit activity that we see during quiet wakefulness disappears during sleep, suggesting that while the hippocampus, the reactivation events themselves are similar, the functions seem to be quite different. And just to summarize that, we see that dopamine unit activity increases consistent with the QTD model of prediction error in the spatial task. Sharp wave ripple units modulate dopamine units, and that this theta rhythm, the encoding cycle for information, can both predict the participation of dopamine units during sharp wave ripple reactivation, and also the structure, the time scale, and content of these reactivation events. It's the unit, it's sort of a unit of encoding, of sequence encoding. And that this doesn't occur during sleep. So sleep, something else is going on. In fact, when we looked at it, I pressed the postdoc to look even harder. Say, maybe there's just fewer sequences, content sequences. Let's just look at the sequences that really have a lot of the really good sequences. Sequences with greater spatial information fidelity actually have lower dopamine content, lower dopamine activation. So dopamine activity is anti-correlated with reactivation content during sleep. Positively correlated during quiet wakefulness, negatively correlated during sleep. And so just overall, we see memory can be encoded during active behavior. It can be replayed in both the neocortex and the hippocampus during sleep. It can be biased, and that the association with reward activity somehow differentiates the functional roles that this reactivation may play during these two states. And I just want to thank everybody that made this possible.
Okay, thank you.
All right.
So we have time for a few questions, and in fact, while the questions are being asked, could we have the next speaker get set up?
Let's do that.
So let's see, the microphones are going around. So here's a question. Why do you think that it's sped up by a factor of, is it 5? The speed with which the replay takes place is...
So actually, initially when we published, we talked about time-compressed reactivation. Sequence took animal 5 seconds to run through, gets replayed in 100 milliseconds. And we estimate these compression factors from 10 to 20. But then this idea that, no, they're actually sequences, these theta sequences. And so what you see is, in fact, there's no compression during replay. There's simply selective replay of the already compressed sequences. So compression is occurring simultaneously. In fact, even thinking about compression, I think, is the wrong way to think about it. That the representation, there is this sort of rapid, sort of time-compressed theta sequence pattern, which happens to, in most conditions, conform to the extended sequence the animal will actually take. But that extended sequence, in a sense, is just a manifestation of the influence those sequences have on behavior. It's not encoded. It's not encoding the long sequence. It's really encoding the short sequence.
Any other questions? In the back. Angela.
Very interesting results. So how does it, it's very clever dopamine system to only replay reward when the hippocampus is replaying a spatial sequence. But how does the dopamine system get information about whether the hippocampus is replaying?
Great question. Yeah, so the question, how does the dopamine system actually know what the sequence that's being played? So there are a couple of possible pathways that we're following up on. One is a pathway that we had looked at previously this had to do with working memory decision making, and that was the hippocampal prefrontal connection. So the prefrontal cortex works with the hippocampus. The hippocampus says, look, this is where you are. Prefrontal cortex says, yeah, this is where you need to be. And so the idea is that you need both of those in order to solve the task. So the prefrontal cortex could have this kind of goal information. It could be biasing that. Another possible pathway, it's a relatively new pathway that's been discovered that goes, it's the one indirect output of the CA field of the hippocampus. CA3 is it's this highly auto-associative subfield within the hippocampus. Lots of interconnectivity. It's where this kind of auto-associative memory functions have been proposed to be expressed, and it has outputs into the lateral septum. And this is adjacent to the hypothalamus. It's been connected with motivation, anxiety, with emotion. So the idea is that that you can have you can direct behavior in two ways. One, it's like, I got a plan. I'm going to do this because I'm going to graduate, I'm going to get a job, and I'm going to get a lot of money. And the other is, I'm going to do this because I'm going to get a reward, right? It's like pulling the lever. It's just, there is this sort of immediate evaluation of probable outcome. And so there could be these two separate pathways. One, long-range, goal-oriented through the prefrontal cortex. One, short-range, sort of emotion, affective-driven pathway through these hypothalamic circuits, both of which converge on the VTA. And so those are the two candidates that we're thinking.
Okay, well, let's thank the speaker again.
All right.
Okay. We're now going to have, hello? We're now going to have two shorter talks. And the first one is going to be by Christine Savin. and Matt Langiel from Cambridge University, together with Peter Dayan, who is sitting in the back of the auditorium.
Right, can you hear me? Okay, good. So we're going to continue talking about memory retrieval, although in much more abstract terms than what we've seen so far. And what we want to understand and how this happens at the level of neurons and of synapses. And what I'm going to argue for today is that correlations in synaptic weights play an important role during memory retrieval, something that has been completely ignored in the previous work. Furthermore, understanding the synaptic dependencies is important because it gives us a new insights into the biological circuitry that underlies this function. Okay, so the starting point is very simple. We have a network consisting of neurons that are connecting through synapses, which we define through their synaptic efficacies. Now, even more schematically, we can look at this in abstract terms. So we can think of neural activity, that is the trajectory, the evolution of the neural activity during over time, as being influenced by the synaptic efficacies. Now, conversely, the neural activity will influence the synaptic efficacies themselves through a process called synaptic plasticity. So the strength of a synapse in this network will change as function of the activity of the pre- and postsynaptic neuron. A simple example of this would be that the synaptic strength changes through a Hebb rule, that is, with a term proportional to the activity of the pre- and postsynaptic, the product of the activity of the pre- and postsynaptic neuron. Right? So in this basic framework, you can think about neural computation as an interaction between these two processes. So for memory, for the memory context, synaptic plasticity corresponds to memory storage. That is, we encode some information about the neural activity in the synaptic weights. Correspondingly, the neural dynamics corresponds to memory retrieval. That is, we try to extract the information that we stored in the weights to retrieve back the original patterns. Right? Now, importantly, virtually all the existing work in computational neuroscience, and one would say even in neuroscience as a whole, focuses on individual synapses in this network. So we're very good at characterizing how the marginal statistics of the synapses depend on the neural activity, but we know virtually nothing about the joint statistics of these synapses. And I'm not sure if you can see anything. Right. So the basic question is, why should we care about these dependencies? And there's a simple answer, that if we look at the experimental data, we find that there are actually interesting synaptic correlations, and we need to understand what this means and how they go back to influence the dynamics of the network. Okay, so for understanding the neural data, what we look at is little circuit motifs. So a pair of synapses that share the same postsynaptic neuron. Now, if we measure the joint synaptic efficacies of pairs of such synapses, and we do this for many such synapses, then we get a cloud of dots of this sort. So we usually find that there are synaptic dependencies between these pairs of synapses. And now this is a cartoon, but we actually have neural data from the cortex, and we find that for a pair of synapses that share the postsynaptic neuron, we have a correlation coefficient of about 0.2. And people have measured this sort of correlations for a lot of little synaptic configurations. They're particularly large in this case when we have a pair of synapses that reciprocally connect to neurons, but they're also there for a pair of synapses that are outgoing from the same neuron or for one of these little chain configurations. Now, so we know that they're there. Now, we set out to answer two questions. First of all, why are they there? How do they come about? And second, what do they mean for circuit function? And in particular, what they mean for memory retrieval. Now, starting for the first question, so far we've looked at pairs of synapses in this network, but what we really want to understand is the joint statistics of all the synapses in this network, so P of W. Now, exploiting what we know from the biology, you can write out a generative model that describes how the synaptic weights come about in this network. So this looks something like this. So we have a sequence of neural activity patterns that each induce changes in the synaptic weights. And if we want to know the distribution of the synaptic weights that what we have to do is we have to marginalize out all these effects of different activity patterns. Now, to understand the detailed dependency structure within these synaptic pairs, what you have to do is to zoom in into this generative model. So again, we're going to look at the pair of synapses that share the postsynaptic neuron. And the generative model looks something like this. So we have the strength of the synapse depends on the activity of the pre- and the postsynaptic neuron. Now, if we look at the pair of synapses that share the postsynaptic neuron, then the history of activity in the postsynaptic neuron acts as a source of shared variability for these two synapses. So naturally, we expect that there should be dependencies between these two. And in general, we expect dependencies between any pair of synapses that share either a pre- and postsynaptic partner. And this is true. It's an effect that is really inherited from the connectivity structure of the network. And it would be the case even if the activities in this network would be completely independent. Now, the specific pattern of correlations that we'll get will depend on the statistics of the activity and on the learning rule that actually operates in the circuit. But we can say that in general, synaptic correlations are expected to be there, and they're really a natural consequence of synaptic plasticity. Now, given this fact, then we have to move to the second question. We have to really understand what they mean for memory recall. And the way that we're going to look at this is we're going to start again with the same generative model. So we have the map between neural activities and the synapses. And now the individual activities patterns, we're going to interpret them as memory items. Okay? So during memory retrieval, what we get is a recall queue, which is really a noisy version of one of the originally stored patterns, and the other ones are remaining here. So what we want is we want to find out which is this originally stored pattern given the information that we have in the recall queue and the weights. Now, if we think about it in these terms, then memory recall is naturally a probabilistic inference problem. So what we really want to know, we want to compute the posterior over any pattern being the pattern that we're trying to retrieve, given the information that you have in the weights and the recall queue. And we can decompose this using Bayes' rule in 3 terms. So we have the prior of what kind of patterns we're storing in the network. We have a noise model that describes how we go from the originally stored pattern to the to the recall queue. And lastly, we have the evidence from the weights. And this is where the correlations will come back into play a role. Note that this is not exactly the same distribution that we had before, but it has a very similar dependency structure because we obtain it by marginalizing out all the other patterns except for the one that we're trying to retrieve. Okay, so now if we look at, we're trying to compute this posterior, we can look in more detail at the evidence from the weights. What happens is that in general, for most learning rules, this has a very complex structure, and it's in general intractable. So what we're going to have to do is we're going to have to approximate it somehow. Now, either implicitly or explicitly, all previous work has assumed a very simple way of approximating this, that is a factorized distribution. So, we're going to implicitly assume that all synapses are independent. Now we can think about this approximation as a first-order maximum entropy version of the original distribution. And if you think about it in these terms, then it's easier to extend this to a higher-order approximation that allows us to take correlations into account. So we're going to construct the second-order maximum entropy approximation of this distribution, where you don't only constrain the marginals, but also the covariance structure from the two distributions. Now, if you want to, if correlations play an important role during memory retrieval, then we expect that the performance will be better when inference takes into account the covariance structure compared to the case when it does not. Okay, so for the remaining time, what I'm going to do is I'm going to look at several representative learning rules that span sort of the favorite versions known in the literature, and for each of them I'm going to derive analytically the first and the second order approximation. Then we're going to plug this in to replace the unknown evidence from the synapses. We're gonna compute the corresponding approximate posterior and derive the corresponding neural dynamics that implement the approximately optimal decoder. Now, due to time constraints, I won't have time to actually explain in detail how we map, how we go back from the posterior to the neural dynamics. This is described in detail both in the poster and in the paper, so we can talk about it later if you want. But this is sort of the general procedure that we will follow. Now, finally, we reached the actual results. So we're going to look at two main classes of plasticity rules that are popular and kind of capture a lot of the relevant aspects of biological neural plasticity. So the first class of learning rules is what we call additive learning rules, where we assume that synaptic efficacies are real valued things, and they change in an additive fashion. So the change from time t to time t plus 1 is incremental, and it depends on the activity of the pre and the postsynaptic neuron. Okay. The other class of learning rule is a lot more abstract. Here we assume that we only have two possible values of the synaptic efficacy. So synaptic efficacies can be either 0 or 1. And we have, underlying this, we have complicated stochastic transitions between latent states describing how you move between these two possible values as a function of the activity of the pre- and postsynaptic neuron. Okay, so this again is very abstract, but it captures an important property of the neural synaptic plasticity. That's the fact that synaptic efficacies are bounded within certain range, and it can't grow arbitrarily large, which would happen in the first for the first class of learning rules. Okay, and the talk following mine will discuss in more detail the memory capacity of this entire class of learning rules. Okay, so the two are really qualitatively very different, and if you find that correlation, that synaptic correlations play an important role in both cases, that suggests that it's really a general effect and is not something specific to a quirk of a specific learning rule that I'm looking at. Okay, so let's start looking at this. The first rule that I'm going to look at is the covariance rule, and this is important because it's basically is the poster child of all the classic work on memory retrieval, and this looks like this. So what we have is we assume that the neural activities are binary, so activities of the neurons can be either 0 or 1, and we increase the synaptic strength whenever the activity of the pre- and postsynaptic neuron are the same, and decrease them otherwise. Okay. Now, first of all, let's look at the correlation structure that is induced by this plasticity rule. So this is actually very peculiar. So we have the weight matrix is perfectly symmetric, so the reciprocal connections are perfectly correlated, but we don't have any other correlations there. And of course, this is very different from what we've seen in the cortical data. So now, for this learning rule, if you ask the question, do we need to take into account synaptic correlations or not? The answer is no, actually. So if we look at the retrieval performance, where we perform a lot of trials and average the obtained error, it's essentially the same. So there's actually no benefit in considering a more complex approximation. In terms of the neural implementation, this is very simple and linear, and in fact, we can rederive the original Hopfield dynamics, which was proposed in relation to this covariance rule, as being the optimal thing for this learning rule. Okay, so the simplicity of the linear dynamics is sort of mathematically appealing, but again, it doesn't really capture the biology of the circuit. There are a lot of nonlinear features in the true dynamics that are not really well described by this. The correlation structure isn't that good either. And lastly, there's a fundamental problem about the learning rule itself. The fact that you get increases in the synaptic efficacy, but when both the pre and the postsynaptic neuron are inactive, and this is something that you never see biologically. So we can do a small change in this learning rule. We can use now the synaptic, the simple Hebbian rule that I described previously, and now the picture becomes very different. So first of all, you get lots of correlations, And at least qualitatively, they sort of match what you see in the experimental data. Now, again, if you try to compare the performance of the decoders that do and do not take them into account, you find a very different picture again. So you're much better if you do take correlations into account, and if you don't, you're particularly bad. That is, you're worse than a control that completely ignores the information in the synaptic in the synaptic weights. So that should be giving us a sort of a upper bound of how bad we're supposed to be doing if our recall is in any way sensible. Okay, in terms of the neural dynamics, we find interesting nonlinearities, and there's an intuitive way to understand this. If the dynamics would be linear, then we treat the synapses as being independent. Now, if there are actual synaptic correlations, then they're really overcounting the evidence from these synapses. So we need additional nonlinear effects in order to compensate for this overcounting. Okay, so now if we look at the second class of learning rules, we consider a particular learning rule where plasticity happens only when the postsynaptic neuron is active, and it either follows the red or the blue arrows depending on the activity of the presynaptic neuron. Again, this models an abstract form of NMDA receptor-dependent plasticity, which is sort of relevant biologically. The structure of the correlations is not quite what we want in the data, but there are non-trivial correlations in there. And again, we find the correlations play an important role during decoding. The performance improvement is not that great, but we can actually compute a lower bound on the estimated performance. So this gives us the expected dynamical range of the error. And the reason this is small is because we've now moved to binary synapses, and there's not much information you can store in binary synapses to begin with. In terms of the neural implementation, this predicts additional interesting nonlinearities in the circuit. In particular, it predicts a nonlinear dendritic integration. So if you measure the current in the neuron as function of the number of inputs that are active, we find a nonlinear dependence between the two, and interestingly, there's similar nonlinearities reported in neurons in the cortex. Now, this is not a trivial result. There are other learning rules from the same class for which we would predict a linear integration of information. Now, to sum up, we generally have a framework where we start from a set of assumptions about the storage procedure, that is the learning rule and what kind of patterns we're storing, and we can derive the patterns of synaptic correlations, whether or not correlations matter for the dynamics, and the actual circuit features that are necessary to do efficient recall. Now, none of the synaptic plasticity rules that we've looked at perfectly matches what we see experimentally in terms of the correlations. However, across the board, we find that synaptic correlations do need to be taken into account, or otherwise we risk catastrophic performance. The fact that the covariance rule is an exception explains the fact that it's so commonly used in the classical work on auto-associative memory recall. And lastly, our model predicts that different circuit nonlinearities that we see in biology can be understood as adaptation for implementing approximately optimal recall in the face of synaptic correlations. And if you're interested in the technical details, our poster is number 61 this evening.
Okay, thank you.
Sorry.
Okay, we have time for maybe one or two questions, and we have the final speaker. Is someone here? Okay, let me.
Hey, thanks for the nice talk.
Hi, Jörg.
If you want, you could see in the experimental data, if you want, 2 clusters. So there could be multiple modes in there. How much more difficult would all your analysis be if you capture more than just a correlation with a Gaussian, but maybe more than one mode?
It's already relatively hard to do the inference even for this case. especially for the bounded synapses. So there is one example of a learning rule where it's not sufficient to look at pairwise correlations, but we haven't really worked out how to go beyond that yet. Yeah, so pretty much all there is.
Okay, thank you very much. Let's thank the speaker again.
Thanks.
The next and final talk of the session is going to be given by Surya Ganguly from Stanford and his co-author, Subhanil Lahiri, which I think Surya will correct me on. But before the talk, this is one of our prize winners, and they receive 2 checks. And here they are.
Thank you. I didn't realize there was money involved. Thanks. Okay, we get one more time. I was thinking that I should do one more time. Try one more time.
So while we're waiting for the computer to connect, I just wanted a footnote for the Matt Wilson's talk. So you remember he said that he thought that this dopamine signal during sleep might be generated through the medial septal nucleus. So there's been an interesting result from human deep brain stimulation of that very same nucleus, and the purpose of it was to try to prevent people from eating too much. And the idea was that that you would stimulate a circuit that would shut down your appetite. And indeed it did, but it only lasted very briefly. But there was a side effect that no one predicted, which was that the subjects that were stimulated, was randomized, could remember better and were smarter. So this deep brain stimulation is something that has been used for Parkinson's disease. It's been used for treating depression, anterior cingulate, and now apparently can be used to enhance and might be very important, for example, for Alzheimer's patients. Okay, we're ready to go.
Okay, thanks. Sorry for the hiccup. So this is a joint work with a fantastic postdoc, Subhanil Lahiri, AKA he goes by Shubi, actually. That's his affectionate nickname. And so he's going to be giving a poster tonight, and he'll be telling you a lot more than I can tell you in a short talk. But let's get started. So this is the New York Times view of the brain. Geometrically, it's wrong in so many ways, but it does capture the important idea that the very basis of our ability to rapidly create lasting memories depends crucially on our brain's ability to modify its synapses in an experience-dependent manner. So this leads to a very natural and fundamental question. What exactly is a synapse from neuron J to neuron I? So the answer to this question reveals an incredible gulf between theory and experiment. If you were to ask a theorist what is a synapse from neuron J to neuron I, they'd say, oh, it's easy, it's just Wij. Or depending on the theorist, it might be Jij. And by what they mean by that is that this analog scalar value denotes the size of a postsynaptic potential injected into the postsynaptic neuron from a presynaptic spike. But if you ask an experimentalist what is a synapse, they'll give you a much more nuanced view, pointing out an incredible plethora of molecular players and many, many molecular signaling pathways underlying synapses. So for example, this is a phosphorylation network of kinases and phosphatases that hides inside every single hippocampal CA3 synapse. All right? So a key question is, what is the functional role of this molecular complexity? What is it good for? How, as theorists, should we confront it, and how should we model synapses? Okay, but actually it turns out that, so the experimental data suggests that maybe we might want to enlarge in our conception of a synapse from a single scalar value to an entire dynamical system in its own right. But it turns out that theoretical considerations also actually demand this, not only suggest it. So if we just think about a very simple question, consider the number of associations a neuron with n afferent synapses can store. So here's a neuron, here's a pattern of activity, a desired output pattern, and here's a time-dependent set of synaptic weights. It's a perceptron-like element, and you'd like to change the weight so it maps this input to this output. So a simple online Hebbian learning rule that could do that is a Hebbian learning plus weight decay to prevent synapses from becoming unbounded. So this actually, the first term allows analog weights to decay slightly, i.e. forget the past. And the second term adds in the new association to the weight, so you learn a new input. And a critical question is memory capacity. How far back into the past can these synapses reliably recall previously stored associations? And the answer actually is if this leak term tau is order n, then the past order n associations can indeed be recalled. So this sounds good, but actually this is completely biologically implausible, because hiding inside the solution is essentially the idea that each individual synapse can reliably maintain order n distinguishable analog states. For example, here you're adding an order 1 quantity to an order n quantity. And so this is completely implausible, because individual synapses have a finite dynamic range and they're noisy as well. So they can only maintain a finite number of distinguishable states. So what about real synapses in which they can only take on a finite number of distinguishable values for their strength? So for example, imagine the worst case scenario where each of these synapses is either strong or weak. Everything I'm about to say generalizes to a finite number of strengths. But so basically, if this is the case, then you can no longer just add an association to the synaptic weights without running into boundaries. So for example, this one synapse could be characterized by 2 stochastic processes, one corresponding to potentiation and one corresponding to depression. If the synapses is weak and external activity pattern suggests it should potentiate, it may potentiate and switch state with a probability Q. But if it's already strong, it can't go any further. And a similar story holds for depression. Okay, so how do these synapses behave? Well, this was first noticed by Stefano Fussi, is that they behave quite poorly. So it turns out that if you make Q order 1, so these synapses are quite jumpy, they rapidly learn what you show them, then the memory capacity is logarithmic in n. So this is an absolute catastrophe. And the intuition for this is each time you write a new memory onto this population of synapses, it rapidly overwrites previous memories, because the population of synaptic switches all flip. One way around this is to slow down the synapses and make the probability of switching extremely small. Then the memory capacity becomes order square root of the number of synapses, but you pay a huge price. You're slow to learn. You have to sort of show a pattern to this population a few times before you can reliably learn it. You can't remember things in one shot. Okay, so basically what this is saying is both experiment and theory are suggesting that we must expand our theoretical conception of a synapse from a simple scalar value, whether it's analog or digital, to an entire stochastic dynamical system in its own right. But this yields a large universe of synaptic models that we have to explore and understand. Right? So we're going to try to initiate that today. So the theoretical approach that we're going to take is the following. So here's a synapse between a presynaptic axon and a postsynaptic spine. And we imagine that the synapse has multiple internal molecular functional states. Some, the red ones, could be strong, and others, the blue ones, would be weak. So externally, there's only 2 efficacies, but there's multiple internal states. Okay? So now a candidate potentiation or depression event, which depends on the pattern of pre- and postsynaptic spikes, can induce an arbitrary stochastic transition between the states. So, and there's m internal states. So the synapse is described by a pair of 2 m by m stochastic transition matrices. If you potentiate, you have one set of transitions. If you depress, you have another set of transitions. And intriguingly, experimentalists are very presciently starting, or have started, to characterize their molecular biology data in terms of these synaptic state transition diagrams. So we're going to be getting more and more of these in the future, and we'd like a theory to be able to generally interpret these and understand what their function is. Okay? So now how do we characterize memory? Well, we imagine that there's a continuous stream of memories that are stored at some Poisson rate in a population of n synapses with m internal states. So this is our population. the memory stored at time t equals 0 demands that some synapses potentiate and some depress, depending on the nature of this pattern being stored. So this yields an ideal synaptic weight vector, W ideal. Okay? However, the storage of future memories after time t equals 0 changes the synaptic weight vector to some other synaptic weight vector. Now imagine that you're an ideal observer that can look directly at the synapses. The degree to which you can recall the memory is going to be related to the degree of overlap between W of t and W ideal. So basically, a way to quantify this overlap is basically a z-scored version of this overlap. So this is W ideal dotted with WT, averaged over all the presentations of future memories, and then z-scored according to the null distribution of what happens at an infinite separation time. Okay? So this is a number that decays over time, so it's a memory curve. And we're not the first to come up with this formalism. This is a well-established formalism in neuroscience. Now, I must say that we're imagining that the patterns are random and uncorrelated across synapses. So we are looking at the case of uncorrelated synapses. But it is the case that this forms an upper bound on the memory capacity, even in the case of correlated synapses. So in particular, when this signal-to-noise ratio falls below 1, no decoder of the past based on neural activity could ever recall the memory, whether the synapses are correlated or not. But it would be intriguing to combine this talk and the previous talk to see what goes on when we really carefully work that out. But we're looking at an orthogonal problem to the previous talk. So now the problem is each choice of N, M potentiation, and N depression yields a different curve. And we'd like to understand the relationship between these quantities in general, right? So all previous work, there's very little previous work actually on trying to understand complex synaptic models. And most of it sort of analyzes one model at a time. So for example, here's a serial model where you have a chain of states and potentiation moves you this way and depression moves you this way. And so this chain has one memory curve, and here's the cascade model of Fusi and Drew and Abbott, and there's a different scaling and a different dependence. But eventually, if we want to have a general theory of molecular complexity and its role in memory, we can't afford to analyze one model at a time. We'd run out of time because the explosion in the number of models as the number of states gets big. So to elucidate the functional contribution of molecular complexity to memory, we want to not simply understand individual models, but understand the space of all possible models within this family. Okay? So we'd like to initiate that kind of a general theory. So the types of questions that we're going to ask and answer in this talk are the following. In general, how does the structure of a synaptic dynamical system, basically specified by these two Markov transition matrices, M potentiation and N depression, determine its function, quantified by its memory curve? Okay? This is a very powerful modeling framework. You can model any system by a Markovian dynamical system as long as you have access to all of its states. So what are the limits of these synapses' power? In essence, what are the fundamental limits of achievable memory over all possible choices of synaptic dynamical systems described by these stochastic systems? And moreover, what is the structural organization of synaptic dynamical systems that achieve these limits? Okay, so this is a very difficult problem, again, because there's a combinatorial explosion in the number of possible models as the number of internal states increases. So what theoretical principles can control this combinatorial explosion in the number of models as M increases? So we've discovered some theoretical principles based on general theories in stochastic processes, and we've actually derived some new theorems sort of controlling this complexity. So let's go through that now. So now the results start. Okay? So the first thing we found is that we were able to impose a theoretical order in synaptic dynamics using first passage time theory. And the basic idea is as follows. The key to understanding the memory capacity of these systems is analyzing the stochastic process that governs forgetting in these systems. So basically, as the synaptic population undergoes continuous modifications due to the storage of new memories after time t equals 0, right, the memory that we're tracking, the internal state of every synapse stochastically wanders around according to a forgetting process. There may be some fraction of times at which it potentiates, and then it'll undergo a potentiation operation, plus a fraction of times at which it depresses, and then it'll undergo, a depression operation here. That's a mistake. So this is another stochastic transition matrix, a forgetting matrix, all right? So this forgetting matrix has an equilibrium probability distribution of state occupancies, right? This is sort of the fraction of synapses in your population in any given state I, okay? It also has a mean first passage time matrix from I to j. So imagine you start at some state I, and you ask how long do you have to wait before you get to state J for the first time, on average, over all possible realizations of your trajectories. That's by definition the mean first passage time matrix. Okay, so what we've discovered is that if we consider this following quantity, right, this is essentially a sum over all the potentiated states. Okay, so if you start at state I, this is a sum over all the potentiated states of the time it takes to get from state I to the potentiated state, weighted by the probability of being in the potentiated state at any given time. Okay, so this is the essentially it's the average time it takes to get to the potentiated states weighted by their equilibrium probability. It turns out that if you order the states from left to right in this order of decreasing potentiation, a lot of remarkable structure emerges. Okay, so just to give you an illustration, here's for example two complex synaptic dynamical systems, both M potentiation and M depression are shown here, and they look like a jumble. I mean, this is what we would get if we measured it from data, if we could do that. Okay, but if we order the states according to this order, it turns out that the structure underlying these models emerges. So this network is isomorphic to this network, and this network is isomorphic to this network. Intuitively, states that are over here take a long time to reach the potentiated states. And so you see you uncover this order. And also states that are over here take a short time to get to the potentiated states. So these are the states over here. And this also gets quickly to the potentiated states. Okay, so this is more than just an interesting graph layout algorithm for these forgetting processes. It turns out that the structure of optimal synaptic dynamical systems are exceedingly simple if and only if the states are placed in this order. Okay? So consider, for example, optimizing a particular quantity, a particular scalar function of the memory curve, for example, the area. We'll discuss other functions later on. So let's consider the space of models that have maximal area under the memory curve. This represents how long you roughly remember the memory. Okay? So we can show that when the states are placed in this order, M potentiation should only have transitions from left to right, i.e. it moves you to the potentiated states. M depression should only go from right to left, i.e. it moves you to the depotentiated states. If you don't have this, then you can find deformations in the space of stochastic processes that increase the area. Okay? So this is sort of intuitive, but the most important thing is we can also find a set of deformations in the space of stochastic processes, M potentiation and M depression, that essentially remove shortcuts. I.e., so let's say M potentiation is a matrix like this. This is some part of it. Everything's moving to the right. But let's say there's some shortcut, skipping a bunch of states in the order that we've discovered. We can find a deformation that reduces the shortcut and increases the transition rates on the direct links. Okay? And we can prove that this deformation, A, preserves the order, B, preserves the equilibrium distribution of the states, and C, increases the area. Okay? So then if we ruthlessly follow all of these deformations, our final endpoint following these deformations is just a chain. And this chain has increased area, or has higher area than the model that we initially started with, no matter what model we started with. So this automatically leads to a theorem that the area under the memory curve of any synaptic dynamical system whatsoever is bounded by that of a chain with the same equilibrium distribution. Okay, theorem number one. Theorem number two, we also showed that the area of a chain cannot exceed order n to the 1/2 m, where remember n was the number of synapses and m was the number of internal states, no matter what choice you make for the transition rates along the chain. Okay, so if you put this and this together, you get the very general result that the area under the memory curve of any synaptic dynamical system can never exceed order n to the 1/2 m. And the optimal system actually is a chain with a particular equilibrium distribution. Okay, but we can go beyond area. Now that we have a constraint on the area, we can get what we actually would like to get, which is what we call a memory frontier, or a frontier beyond whose bourne no memory curve can cross. So basically, this is so in the SNR time plane, we've derived a curve, this green curve, and we can show that no matter what memory model you choose, the memory curve of that model can never enter this forbidden region. Right? Every memory model will have a curve somewhere in this region. And at late times, where the time in the inverse rate of plasticity events is larger than the number of internal states, this memory curve is simply a power law with exponent -1. And it expands as the square root of the number of synapses, and it expands linearly in the number of states. And this is sort of numerical evidence that our proof is correct. Okay? So which models touch this memory envelope or memory frontier? Chains with different transition rates come close to the frontier, especially at late times. And so that's what, and these chains have memory curves that touch the envelope somewhere, but are worse at other points. And depending on how you optimize a chain, you get to the envelope at different points. So the family of chains traces out this envelope. Okay? Now, there's a lot more to say about this, and Shubi will talk about it in the poster, but it's intriguing to note that various measures of memory, like the area, the coefficient of the frontier, the lifetime of memories at which the SNR drops below 1, they all grow linearly with the number of internal states, but grow only as the square root of the number of synapses. So it's much better to have a small number of complex synapses than a large number of simple synapses. And beautiful work by Seth Grant at Cambridge has actually shown that not only have brains gotten bigger over evolutionary timescales, synapses have gotten more complex. So this theory makes the intriguing sort of speculation that potentially the elaboration of synaptic complexity that has indeed occurred over evolutionary timescales could potentially be could potentially yield a strong contribution to our long-term memory storage capabilities. And so we should take maybe synaptic complexity more seriously. I'd like to end with just a sort of a perspective/speculation. Understanding synaptic complexity could potentially have lots of different dividends. On the neurobiology side, it could give us a framework a theoretical framework for imposing order on molecular neurobiology data. We've actually already started to do this. Under review, we have a manuscript describing cerebellar learning using complex synaptic dynamics, and we show that for the particular data sets collected from Jennifer Raymond's lab at Stanford, we need complex synaptic dynamics to explain the data. On the mathematics side, it can yield dividends about new theorems about perturbations to stochastic processes. In fact, we've actually proven tighter bounds on specific classes of perturbations than any bounds that we're aware of in the literature. So there's actually some new theorems hiding in there. And probably for the NIPS crowd, an interesting speculation is that synaptic complexity may actually play a role in the next generation of artificial neural networks, allowing us to do things that we might not have been able to do before. We have several hints of that already. So for example, solving spatial temporal credit assignment problems, well, these can partially be solved by putting sophisticated memory and decision-making capabilities inside synapses. And also, recent work by Nicolas Brunel and collaborators have shown that if you want to learn if you want to do supervised perceptron learning, for example, with discrete synapses, it's useful to have a set of hidden states. And the dynamics of the hidden states can be described through or derived through message passing algorithms between synapses and neurons. And so we've probably hit the tip of the iceberg in what we might be able to do with synaptic complexity. So I'd like to just throw out the speculation that artificial neural networks with more complex synapses may represent sort of a new type of deep network where the depth is now placed at smaller and smaller spatial scales within synapses rather than across multiple layers of simple synapses. And so I think there might be a lot to do here. Okay, let me end with acknowledgement. So again, I'd like to thank Shubi. He's a fantastic postdoc. He will tell you again a lot more than what I could tell you tonight. I encourage you to visit his poster. I'd like to thank various people for very interesting discussions and my funding. Thanks.
Okay, we have time for a few questions. We have one right here in the front.
So to what extent does this theory predict time-dependent synaptic plasticity?
Oh, they're almost, you can't dissociate it from that. So basically, whenever a synapse has internal states that you can observe through the lens of extracellular physiology, that endows the propensity for potentiation and depression to depend on the prior history of potentiation and depression events. So metaplasticity and synaptic complexity go hand in hand.
Okay, another question here, and then we're going to go on to the spotlights. Please take the microphone.
Hello. Lovely,
hey.
lovely intervention. Have you looked into understanding how sparsity in the patterns is actually influencing your the theorem you actually?
Yeah, we haven't done a deep study of that. That's on our to-do list for sure, definitely. Yeah, I know you've done some nice work on that.
Okay, let's thank the speaker again.